Okay, so we finally get to talk about cells. At this point, we've covered quite a bit of chemistry. And again, the whole purpose of covering all that chemistry was to learn about the four principal biomolecules of life: the carbs, the lipids, proteins, nucleic acids. And now we're going to put them all together to actually create a cell so that we understand basically what a cell is and how it functions. All right, so let's get into this cell structure and function. First thing we're going to cover are microscopes. Then we'll discuss cell theory, one of the two important theories of biology, the study of life. And then we'll get into all of the structures of cells, the organelles basically, and what do each of them do, all right? Okay, so what you're looking at here is a size scale of life and or molecules. So notice that we spent quite a bit of time talking about atoms, right? And then building these biomolecules that you see here, right? Lipids, proteins, and so on. Carbohydrates can actually be super big, like starch. When you get into lab, you might see starch granules under a microscope. That's how big they are. You can see them with a microscope. Imagine that, seeing a molecule with a microscope. You can see also that viruses are really not that much bigger than these molecules, and that's because viruses are basically made up of like protein and some sort of DNA or RNA, right? So they're actually just, more molecules and not considered to be alive. And then we will start seeing organelles, right? Like mitochondria, right? Nucleus. And somewhere in between that, mitochondria and nucleus are things like bacteria. So if we wanted to see any of these, whether it be molecules or tiny parts of cells, there are very specialized microscopes we would need, like an electron microscope might be helpful, looking at internal parts of cells and maybe large molecules. In lab, we work with a light microscope, right? And that allows us to see cells that are in the micrometer range. You might remember the micrometer, that funny looking mu micrometer. So light microscopes are scaled to be able to see the micrometer range, which is the size range that cells are in. And also there are some cells that you can see with the unaided eye, like an egg is a cell, right? It's a chicken egg. Eggs are cells. And then there's like the anomalies, like the really massive neurons, as you might find on a giraffe, right? But clearly we are in the size range of meters, and so we can be seen with the unaided eye. Okay, so the idea is to focus on the light microscope because that's what allows us to see cells and their internal parts. So here's what I propose that we do. We can spend time talking about each one of these types of microscopes. But at some point, we should probably make a table that allows us to see the differences, like what are the benefits of each and what are the drawbacks of each, right? So very quickly, the light microscope is the kind of microscope that we use, but the electron microscope is significantly better. Still, the light microscope is very popular today still because light microscopes allow you to see living cells, whereas electron microscopes require those to be basically killed. So you're not looking at them live. They're not in motion. You don't get to see behaviors, but you get very detailed images from electron microscopes. The light microscope uses light. Remember, I'm going to go through these slides, but then at some point I'm going to create a table so that we can compare and contrast light microscopes from electron microscopes. We're going to compare light with electron. All right, so the light microscope uses light as an energy source, and it can only magnify up to 1,000 times. You can actually magnify higher than 1,000 times, but the problem is that you lose resolution above 1,000-fold magnification. Okay, so above 1,000, we lose resolution. And resolution is what you call clarity. It's the same thing. So it turns out that the light microscope is only good up to 1,000 times, because if we try magnifying above that, which we can, we lose clarity, and at that point we're not getting any valuable information. So we don't bother going over 1,000 times. Now, the light microscope will allow you to see living cells. Remember, that's a massive advantage to it. But you can get greater magnification with an electron microscope. The electron microscope uses electrons to supply the energy, not light. And it can magnify a whole lot more, like 100,000 times. So you can say it's 100 times better than a light microscope, and that's a big deal. The problem again is that when you use an electron microscope, uh, you can't see living things. Um, and overall, no matter what microscope we're talking about, there are two important features of a microscope to know. Magnification is one. Resolution, however, is the bigger deal. That's clarity. That sets the magnification limit. All right, so what do cells look like when you use a light microscope? Here's a cell called a paramecium. You don't need to know the name of this cell. I just am going to write it down. Paramecium is, as you will learn, a eukaryotic cell. It's not a bacteria. It's a single cell eukaryote. It has a nucleus, and that's what makes it different from bacteria. We have cells with nucleus. That's why we are not bacterial cells. And you can see that this paramecium has some stuff inside, like these bags, and some grainy material in here that you can see all throughout. And then you might also notice that there's some fuzziness on the outside of the cell. That line that you see dividing the inside from the outside, that would be the cell membrane. And cell membranes are made out of phospholipids, you might remember. See, a cell is chemistry. All right, so that's pretty much all we can see with a light microscope. However, there are ways to adjust the light microscope so that you get to see more things, usually at the expense of seeing other things. Here's what I mean. This is the same exact cell. But do you see how dramatically different it looks? This is still a light microscope. It kind of looks like some sort of Photoshop thing, but it isn't. It's a light microscope that made that. And what we have done here is we've decided to get to the bottom of what is that fuzzy stuff outside the cell. And it turns out to be something that looks like hair. You're going to learn that this is called cilia. I'm going to write it here, but you're going to see it later in the lecture again. Cilia are like hair-like proteins. Cilia is made out of just protein. And not surprisingly, you learn that protein can be the thing that our hair is made out of, right? And so here's cilia on the surface of a eukaryotic cell, like basically extending out of the cell membrane, and it's their version of hair. Also, do you notice that the cell membrane kind of looks like a net? It's not smooth, right? Like there's some sort of texture to it. It looks like a net. All right, so what are we looking at? We're looking at a light microscope produced image of cilia where we have used fluorescence and specifically focused on the hair-like structures on the outside, but we can no longer see the details of the things that were inside. But we could, if we wanted to, we can adjust light microscopes to help us see different things. We can use stains, like you're going to use in lab. Uh, we can also use fluorescence. We can use antibodies. We can get really, really crafty pulling out information about cells. Okay, but still, what you want to know is that the light microscope can only like give you limited magnification because we're using light as an energy source, and it's not as strong as electrons as an energy source. And the single most important advantage to the light microscope is that it allows you to see living things, right? But it peaks at 1,000 magnification because anything above that, you lose clarity. Okay, the electron microscope, on the other hand, is like powerful. It magnifies 100,000 times. So what can I do to help you understand the reason for this? Okay, when we talk about energy, a physics concept. Energy travels in waves. And when light is used as energy, let's say that this is the specimen we want to look at. It's just a little grainy looking thing. Light travels like this. You see what just happened? The light energy just went right past that little grainy thing and it never touched it. The smaller the substance is, the harder it is for light to see it. But what if we were trying to look at an insect? Well, yeah, the light is going to hit it because the insect is much, much larger and will interact with light like better. All right, an electron microscope, however, uses electrons. And electrons have like a super tiny wavelength. So I think you know what's going to happen. It's going to hit the specimen, it's going to bounce off, and that's what's going to give us information. Just like if a light wave were to hit the insect, it bounces away and gives us information. So that's the reason why light is just not going to give us the same level of detail as an electron microscope. Electron uh, energy travels in a much tighter wavelength. Here I'm drawing it as best I can, right? And so it's definitely going to hit very tiny uh, specimens, whereas the light microscope is going to go right by them, never even touching them. Okay, the electron microscope is interesting. There are way more sophisticated looking electron microscopes, but I like to use these images because this is what electron microscopes used to look like back in the days. To some extent, they're still used today, right? But check this out. I like to show what a computer used to look like. And that's a keyboard over here. This is wild stuff. Look at this computer back there that the technician is using. This is the actual microscope here. That's the microscope. These big, like cylindrical, tubular microscopes. And if you notice, there's a big cable behind each one. So looking at the one on the right here, that cable, I'm going to darken it in. That cable is delivering a high amount of voltage. And you can see a very big cable also on the left microscope as well, delivering high voltage. It's basically the source of the electrons that are going to rain down onto the specimen. That voltage goes to a tungsten filament, and tungsten as an element releases a bunch of electrons and rains them down onto the specimen. And this is the sort of image you can generate. Notice that there's no color to them for the most part, right? They're basically black, white, gray. And that's usually how you know you're looking at an electron microscope. If you're looking at color, that's usually better preserved with a light microscope. So colored images are basically light microscopes. Black, white, and gray images are made with an electron microscope. On the upper right, you're looking at pollen. That's one way pollen looks. There's a variety of different types of pollen out there. This is a very common looking pollen. Notice that it kind of has a golf ball kind of uh, design. And that golf ball design allows it to make better use of wind currents so that it can travel farther, just like a golf ball. Life figured it out before we did. On the upper left, you're looking at the compound eye of a fly. Every one of these little circles right here, every one of those is an eyeball. So the whole thing is just like a population of eyes. And notice how um, like fuzzy slash hairy the insect is. And then notice all these structures coming out of the eyeballs of flies. You didn't know eyes that flies were hairy? That's how they know that you're coming for them when you try to swat them. When you try to swat them, your hand above a fly coming at it is pushing air towards the fly, and the fly feels the air with all of that hair. And so it just goes with the wind current and it gets away from you. It knows when you're coming because it's sensing the air you're pushing towards it. Okay, Down below the eye of the fly is an ant. You probably figured that one out. And then on the lower right, that's a flea. Super elegant with that cape. Okay, so those are all like images generated with an electron microscope. That's insect booty. I don't know. I thought you might be curious. What does an insect butt looks like? There it is. I suppose you can look at it as much as you'd like. Go back to this, pause the video or whatever. Uh, I'm just like wilded out by this insect booty. It's hairy. Anyway, this is a worm, and this worm is interesting because of where it was discovered. It was discovered deep in a hydrothermal vent. This is way deep down in the ocean where pressure is super high and where the temperatures are really high also. So the pressure and the temperature would have made it such that life could not exist there. But looks like life found a way. And what's even more interesting, perhaps, is that it has a mouth and it seems to have its version of teeth. This thing is microscopic. And if it has a mouth with teeth, that must mean that it's eating something, right? So there's other stuff down there. Very cool. Okay, so let's do a comparison of the two types of microscopes that I've discussed, right? All right, so we'll compare the light microscope versus the electron microscope. And then we're going to discuss two types of electron microscopes. All right, so first we talked about the energy source, right? So the energy is going to be easy. This is light. These are electrons. Okay. And then we can also look at the strength of magnification. This one goes up to 100, or I'm sorry, to 1,000 times, but this one is 100,000 times. And that's because, again, it maintains resolution. What else can we compare? How do you focus an image? This one uses lenses to redirect light where you want it to go. The electron microscope instead uses magnets to redirect electrons. Electrons have a charge, you might remember, right? They have a negative charge. So we're going to use magnets to redirect the flow of electrons. And finally, we can look at the specimen itself. And remember that this one can use a specimen that's alive, whereas the electron microscope, the specimen is dead. So this is basically why we still use light microscopes, because looking at life in action is also super informative, right? Not just looking at the details of life. All right, final comments on the microscope. There are two types of electron microscopes. One is called scanning. The other one is called transmission. So I'm going to go back and show you. This is a scanning electron microscope that took those images. We were scanning the surface of these insects and pollen. Scanning the surface of that butthole. Scanning the surface of this microscopic worm. All right, so those are scanning images. What we're going to be seeing a whole lot of is going to be transmission. Transmission is when you're looking inside of the cell. You're not just scanning the surface, you're looking inside of it. So we're going to be seeing a lot of these type of images as we go through the lecture. All right, so we're done with the microscope. Let's go on to cell theory now. There are two things you want to know about cell theory, and you're going to be asked about this on the exam. First, you need to know that all living things are composed of cells. Whether it's one cell or many cells, all life is made up of cells. And the second thing you need to know is that all cells come from other cells. Cells give rise to other cells. So if you're going to see cells under the microscope, and you know that all of life is made up of cells, and you know that cells come from other cells, why is this still called a theory? Why isn't it called a law? All right, let's talk about theories. Theories are growing bodies of knowledge. So we're not done learning about these things. As much as we know about cells, there are still some things about cells that we don't fully know and understand. And so until we do, we're going to continue calling this cell theory, right? That's one reason we call it a theory. Here's the other reason. If all cells come from other cells, right, you can keep going back in time. This cell came from this cell came from this cell, and that cell came from this cell, and this cell came from that cell, and so on. You can keep going way, way, way back in time. At some point, you're going to have to address what question? You're going to have to address what was the original cell? Like, where did life begin? And that is a much more complex question to ask. And that's likely the reason why this will remain a theory for a while. When we figure this out and we figure out all the other aspects of cells, then maybe we can start discussing this as the law of cells. But right now, it's a growing body of knowledge, and that's the definition of a theory. Okay, try out this checkpoint question on your own. Which type of microscope would you use to study A, the changes in shape of a living human white blood cell? Okay, the operative word there being living. So what's the cell we want to use there? I'm sorry, the microscope we want to use there? We want to use the light microscope, right? And let's do part B. Which microscope would you use if you wanted to look at the finest details of the surface texture of human hair? Surface, that's going to be scanning, right? Scanning surfaces. And if you wanted to look at the detailed structure of an organelle inside of a liver cell, that's when you're going to use transmission. That's the most detailed way to look at stuff. It's got to be with a transmission microscope. Okay, hope this is kind of like making sense to you, at least with microscopes, and then cell theory. All right, let's talk now about cell sizes. So why are cells in the micro range? Why aren't they bigger? Why aren't they smaller? Okay, there's two reasons to consider here. One, why aren't they smaller? Okay, cells cannot get any smaller than what we see because we would have to get rid of organelles if we wanted to get smaller. And it turns out that, as an example, if we wanted to make you microscopic in size, or if we wanted to just make you tinier, right, where your organs cannot get tiny, I think you see the issues, right? You need to have a certain size liver, a certain size heart, a certain size brain, a certain size digestive system, and so on. So to try and get smaller would mean having to get rid of those parts, and that's the issue. So it seems like we cannot get smaller because our organs, right, need to be of a certain size. Now you might be wondering, well, why don't the organs themselves get smaller? Okay, the organs can get smaller, but then in the case of a cell, when those organelles get smaller, they're not going to be as efficient at accomplishing all of the chemical reactions needed to sustain life, even if that cell got smaller. So the idea is that cells cannot get smaller because they absolutely need the amount of or the size of their organelles to accomplish all of the activities inside of a cell in an efficient way. Why can't cells get any bigger? If we know why they can't get smaller, why can't they get bigger? And for that, it's definitely all about efficiency. So, how do we measure efficiency for a cell? All right, so imagine a cell is big like this versus small like that. I think we can agree that the larger the cell is, the more uh, resources it's going to need, right? It's going to need more energy. And also, it's going to produce a lot more waste. The smaller cell requires less energy because it's smaller, and it's going to generate less waste. But here's the thing you want to understand. The the outside here, this membrane, the cell membrane, is making contact with the environment here, 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 all the way around, correct? And also remember that the cell is not like a circle. It's a sphere, right? So it's making contact all around. Imagine like a ball. It's making contact with the environment all around that, right? So it has a very high surface area. And the smaller cell is making contact, but it's got less contact with the environment because it's smaller, so it's got a low surface area. So I'm just drawing arrows to indicate high versus low, right? High surface area, low surface area. Okay, but as the cell gets bigger, the volume gets massive. So now I'm using like double arrows to indicate that the volume is like way, way big now. And um, with the cell that is smaller, the volume is also small, but not tiny, tiny. If you were to put numbers to the arrows, here's what would happen. This would give you a small number, and this would give you a large number. And the larger the number, the greater your efficiency. So we can try it. We can actually put some number values here. Let's pretend that the cell is the shape of a cube. And it's like 3 units across and 3 units tall. And because it's a cube, it's got 6 sides. All right, so what's 3 times 3 to figure out the surface area? You would just go length times width, right? Or length times height. 3 times 3 is 9. And because there are 6 sides, that's going to give you a surface area of oh, I'm sorry. I meant to say over here. 54. So from here to here, that side of the cube, that's got a surface area of 9. And if you multiply it by the 6 sides that it has, right, that's going to give you 54 units, whatever the unit is. Centimeters, meters, millimeters, micrometers, more likely. It's going to be 54, right? That's the total surface area. Now, what is the volume? Well, volume is length times width times height. So that's going to be 3 times 3 times 3, right? 3 times 3 is 9 times 3 is 27. And now if you take the surface area, 54, and divide it by 27, you get a value of 2. That's the surface area to volume ratio. Surface area divided by volume gives you a ratio, and in a case of a large cell, it's small. Small value. All right, what if we instead took the big cell and we broke it up into tinier cells? Instead of being 3 across and 3 tall, it's now 1 across and 1 unit tall. Okay, well in that case, you still have the same volume, 27, but your surface area is now like more than doubled. Because now you're taking the surface area of that guy times 6, that guy times 6, that guy times 6, and then like multiplying it by all the cells inside. So now it's 162 divided by 27, and that gives you a value of 6. It turns out that this is way more efficient. It's more efficient to take all the volume and break it up into tiny cells, and those tiny cells are going to give you a lot more efficiency. So you're going to see examples of cells using surface area to volume ratios. So let's make sure that we really pay attention to that. When you take the surface area and you divide it by volume, that gives you a ratio. Surface area to volume ratios are really important to living things. Okay, so how is it that we see evidence of high surface area to volume ratios being so important to life? All right, here's a series of images to help us understand it. Let's begin with the elephant in the upper right. The elephant has massive ears. And if you were to describe those ears, can we agree that they have a lot of surface area? They make a lot of contact with the environment, right? But what about the volume? Are these thick ears or are they very thin? They're super thin. Super thin. So thin, in fact, that if you were to look behind the ears of this elephant, the backside, they look bright red. And that's all the blood flow going to their ears. That's the way an elephant is trying to keep cool. It sends hot blood to its ears and then it fans the ears to cool down the blood. And so the blood that returns back to the elephant is cooler. That's the way the elephant cools itself down. And the ears are like super thin to ensure that it's a very efficient transmission of like energy. Um, on the upper left, I have the image of a dog. Maybe I'll use red to make it pop a little bit. If you follow with me, here's the lip of the dog in red. And then right about here, I was trying to show that there's like these little wiggly things you see very commonly in dogs. The lower lip tends to like flare out and a whole bunch of hot saliva drips down on a hot day. And that hot saliva is kind of dripping off of those weird wiggly things. All right, so these are two high surface area to volume ratio strategies that life uses to make things happen more efficiently, to make things happen quicker. One thing that life does is it makes things super flat and broad, that the elephant is giving you that example. The other thing that life does to make things really efficient is it creates these wiggly structures. And the dog lip in red is showing those wiggly structures. It turns out that hot saliva drips off faster when you have those wiggly structures than if you didn't have them. So it cools down more efficiently. Okay, here's another example of wiggly structures. You're looking at it in the lower left hand. You're looking at the small intestine of an animal, like a human. All right, so the small intestine for us, right, is the longest portion of our intestine. And if you were to cut the small intestine and look into the tube that makes up the small intestine, you would find like a smooth muscle layer on the outside, some mucus producing kind of layer, and then you would see the layer that actually absorbs nutrients and it's wiggly like that. And if you were to like fillet it and open it up, you would have smooth muscle, mucus producing stuff, and then that wiggly stuff. And that's what you're seeing here. That's all this wiggly stuff. Those are called villi. You'll learn about those in anatomy, those of you moving on to that class. And the reason why they're important is because it allows us to absorb nutrients all along the surface of that villi. This is what makes our small intestine so efficient at absorbing nutrients. Okay, can you imagine what that intestine would look like if it did not have the villi? If you just fillet it open and it didn't have villi, it would just be that. So you would be absorbing nutrients where? Just there. That's not efficient at all. Look at all of the nutrient absorption you are doing with wiggly intestine versus if you didn't have that. So that's efficiency. Again, life is going to show you that it wants to be efficient whenever it shows you, right, these sort of uh, folded membranes. I'm describing them as wiggly. Or when they're showing you flat and spread out kinds of membranes. And finally, we also make use of surface area to volume ratios, like when we create things like towels, right? How do you know a good quality towel from a poor quality towel? Well, it's the same way we measure the quality of sheets and so on. We're looking at something called thread count. Like literally, how much thread do you have? So you have a lot of thread. Wow, that's going to be plush. And that's going to be excellent at pulling moisture off your skin when you get out of the shower. That's an awesome towel. And then there's a towel that, your family that you visited give you, and it's like, it's got gaps in it. You know what I'm talking about? It's like an old towel. And you're like, what in the hell? This is not going to dry me. It's going to chafe my skin, right? That's a terrible towel right there. That's garbage. That's trash. Give me that. That's a plush, soft towel. That's going to be awesome at absorbing moisture because there's so much thread to absorb the moisture. Now I want you to think about the towels that we have in the lab and how terrible they are. You know the brown ones that we use, the paper towels that we use from the dispensers? They're terrible. Why? Because they're flat. Compare those to the towels you have in your kitchen. The ones you have in your kitchen are like made out of 2 layers. They're 2-ply, and they have depressions in them. They have this. And the towel isn't just like a straight thing like the ones in the lab. Your towels in your kitchen do this stuff. You see the effort to make wiggly stuff? That increases the absorptive power of those towels. Okay, we've discussed now high surface area to volume ratios to help us explain why cells are tiny. They are tiny because they're trying to be efficient. And do you see the wiggly stuff in the mitochondria? That's for efficiency. And you're going to keep seeing that in a lot of the structures that we look at in cells. Okay, let's start looking at cells, and we're just going to take a look at the bacteria, and then we'll do a second lecture for the eukaryotes. All right, so what you're looking at right now is an example of mostly lipids, right? You can see the lipids here that we talked about. These are specifically phospholipids. You can see the dark gray. I'm shading it in blue. That's the phosphate head. That's a functional group. And then you can see that it's got 2 fatty acid tails sticking out. All of them do. Those are phospholipids. And there's a small ocean of phospholipids. Just a ton of phospholipids. So our cell membranes, any membrane, is made up of phospholipids mostly. What is the difference between one membrane and another? It's the kind of proteins that are on them. Proteins in here are drawn in like a purple color. Those proteins do very specific things. Proteins are very specialized workers. So the proteins you find on the cell membrane of a cell are different from the proteins that you find in other membranes of the cell. And that's why the membranes do different things, because they have different proteins on them. Also, do you see the terms hydrophilic and hydrophobic? Remember, hydrophilic means water-loving. Hydrophobic means water-fearing. The heads are facing the water because they have charges. So those charges interact with water. Remember, water is charged. But the tails, the fatty acid tails, they are hydrophobic. They are fatty. They don't mix with water. So they move to the middle. And that's why you see the fat always in the middle and then the gray heads on the outside. Okay, those are membranes. And all cells that we talk about, all of them have membranes. All of them have a cell membrane. All cell membranes look like what you're seeing right now. Okay, let's talk about our first actual cell. These are prokaryotic cells. This is bacteria. So I'm going to write here prokaryote. A prokaryote is a cell without a nucleus. And the only example of prokaryotes in the world are all bacteria. So when you say prokaryote, it's like saying bacteria. When you say bacteria, it's like saying prokaryote. They're one and the same. So, but a prokaryote is defined as not having a true nucleus. So no true nucleus. The nucleus is where cells store their DNA. But even though bacteria have DNA, they don't store it in a nucleus. They just throw it in the middle of the cell somewhere, and we call that the nucleoid region. In fact, I don't even use that as a term. I think we have enough terms to worry about, so I don't even bother with that term. They don't have a nucleus, so it's just DNA, but not inside of a nucleus. Okay, let's start on the outside and work our way in with this particular bacteria. We're using Helicobacter pylori, which is implicated in stomach ulcers and also like stomach cancers. Also, notice that this is the cartoon image over here, but over here we're looking at an electron microscopic image. And this is a transmission electron microscope. You can see the acronym there. And it's magnifying the image 20,940 times. This is like uh, 21 times greater strength than a regular light microscope. All right, starting on the outside, moving in. Let's begin with flagella. Flagella are used by bacteria to swim through water and position themselves in a better environment, a better temperature, something that has nutrients, um, something that's a little bit more fluid. Bacteria can have flagella. Do all of them have flagella? No. Some do, some don't. In fact, those of you going on to microbiology, you're going to be learning about bacteria in part by way of whether or not they have a flagella or not. You're going to see evidence. You're going to have tests that allow you to determine if your bacteria has flagella or not. You're going to be able to see that it moved or it didn't. Okay, flagella. Flagella is used for moving through liquid environments. We are going to use a term to describe that movement. We're going to call it motility. Motility is just moving through liquid. Okay, next, from the outside moving in, fimbriae. Fimbriae over here in blue. Those are those little hair-like structures. They might remind you of cilia, the way you saw them in paramecium, but they are totally different. They're not the same thing. Paramecium you're looking at here is a eukaryote. It has a nucleus. And the cilia are made up of a different kind of protein than fimbriae. Fimbriae is a different protein. So what do fimbriae do? Fimbriae are used for attachment. So sometimes bacteria get to a place where they like it, and so they want to attach and stay in that spot. So they use fimbriae for that. Um, okay, so that's for attachment. Okay, next, moving in. In yellow is the capsule. And the capsule is used by bacteria for protection. They use it for protection against white blood cells. Those are the cells of our body that protect us against pathogens. So, if a bacteria has a capsule, that usually means that it's going to be a difficult thing for our white blood cells to take care of. Capsules are a big deal in the clinical field. Capsules also allow bacteria to create what are known as biofilms. Biofilms would be that a film of bacterial growth on, let's say, the surface of your teeth. Those are biofilms. And they can create issues for medications. Biofilms protect bacteria from treatment, from medications that we use against them. So they are very much a protective feature for bacteria. But I want you to know that they're used for protection against white blood cells. Okay, moving in is the cell wall. All right, bacteria have a cell wall, and they use that also for protection. The vast majority of bacteria have a cell wall. It's rare to find a bacteria without a cell wall. Capsules, on the other hand, some have them, most don't. Flagella, some have them, most don't. Fimbriae, similar. But when it comes to cell walls, the vast majority of bacteria have the cell wall. So we expect you to have a cell wall. In microbiology, we can test what kind of cell wall you have. There are two very basic types of cell walls that bacteria have. They have a thick cell wall, they have a thin cell wall. We call them gram positives and gram negatives. And that's one of the very first tests you use in the lab to determine what kind of bacteria you have. Okay, so cell walls are used for protection, but they're not for protection necessarily against white blood cells. It's against bursting open or like blowing up when you take in too much water. So I'm going to write here from too much water. All right, cell wall is for protection against bursting if you should take in too much water. So imagine like blowing up a balloon with air and then keep blowing up the balloon. Eventually, the balloon will reach its stretching capacity and it'll burst. That's what happens to bacteria if they take in too much water. Now imagine taking that same balloon and putting it in a shoebox and trying to blow it up. It'll never reach its max because the shoebox is preventing you from doing that. A cell wall is like a shoebox. Okay, moving in is the cell membrane. Here it's described as a plasma membrane. I like to call it cell membrane. So cell membrane just reduces the vocabulary. Plasma, you don't need to worry about it, just cell membrane. The cell membrane is basically that, right? It's the outer membrane of a cell. Here's another cell membrane we saw earlier, right? In blue, that's the cell membrane right there, all the way around, okay? All right, so all cells have a cell membrane. Okay, we've covered the outside of the bacteria. Let's move in now. There's only two things inside of bacteria. Ribosomes, those little grainy brown dots. Ribosomes are used to make protein. So ribosomes build protein. And they do it using RNA instructions. You might remember that from the nucleic acid biomolecule. DNA is used to build proteins, but you need to first make a pit stop through RNA. All right, that's ribosomes. And finally, there's the DNA, right? This blue wiggly stuff inside, that's the DNA. And the DNA, again, is instructions for building protein. And not surprisingly, you have ribosomes that will take the instructions in RNA form and they will build more protein. Okay, that was bacteria. Bacteria oftentimes are described as being simple, but I would caution you in thinking of them as simple, right? They are super efficient. They're little baddies. They reproduce quickly. They get through life super efficiently. And that's what makes them still very successful today. And they're no joke when they cause infections in us. Okay, here's the shapes of the bacteria you're going to be seeing in lab. The smallest bacteria are those circular ones there. Those are called cocci. You might have heard of a streptococcal infection. Staph aureus, one of the causative agents of acne. They're those tiny spherical ones. These rod-shaped ones over here, those are called bacilli. They have the shape of a rod. E. coli, you might have heard of E. coli. That's a bacilli-shaped bacteria. And E. coli can have a flagella also. And then over here, the spiraling one, we call that spirilla. I think you can see why. Syphilis is a spiral bacteria. Vibrio cholera is another one. Okay, so you're going to be looking at those shapes of bacteria in the lab, 3 different shapes. Okay, let's look ahead at what the next lecture is going to look like. In the next one, we're going to cover the eukaryotic cell and look at all the stuff inside of a eukaryotic cell. It's got a nucleus, that's what makes it a eukaryote. And then look at all the wiggly membrane inside, like right here. Maybe use something in red to make it pop. Oh, let's try that. Okay, yeah, that works. Look at all that wiggly membrane. Even that over here, the mitochondria. All of them trying to show you like massive efficiency in whatever it is that they're doing, right? This is why we covered surface area to volume ratios. Every one of those structures I've circled in gold are showing you high surface area to volume ratios. And what you're looking at there is an animal cell. And we know that because there is no cell wall. As soon as you put a cell wall in there, see how it has a boxy shape? That's a very thick cell wall. If you have a cell wall, you have massive protection. Okay, we'll see that in lecture part 2.